Alright, so in this video, I'm just going to be going over downloading a human coronavirus protein set and searching for replicase polyprotein 1AB in the protein set that we download, the human coronavirus. And then I'm going to be using some command line software such as Hammer and Muscle. And the particular data that we're going to be using is, as I said earlier, the human coronavirus protein set, and we're also going to be using annotated and reviewed bat replicase polyproteins from UniprotKB. And then how we're actually going to do this is that we're going to take those bat replicase polyproteins and we will build, using Hammer, a profile hidden Markov model. And once we build our profile hidden Markov model with the bat replicase proteins, we will use that to search for other replicase proteins in the human coronavirus protein set. And then I have here some general steps that I'm going to be doing. I'm going to download the software first, gather the human coronavirus genome, and I have a link here. Here's the, I think the nucleotide core session number right here. And then step is actually we can gather up the bat coronavirus proteins. Now we have Muscle here because Hammer Hammer is a software suite, so it has a few different commands that we can use once we download it. And one of the commands takes in a multiple sequence alignment and builds a profile hidden Markov model. And so we're going to first gather, once we get to the replicase proteins, once we get those, we're going to use Muscle to make a multiple sequence alignment, and then we're going to use Hammer to build the profile hidden Markov model. Once we have that model, we're going to search with that model against the human coronavirus protein set that we download and search for any similar replicase proteins with the bat replicase proteins that we got. So I'm going to go ahead and get started. This is just a general, I'm just going to just go about, and this is general steps. I might skip around here and there, but my main goal to get to you is that we're going to be downloading software. I want to show you how I'm going to download the software, how do I use the software from anywhere, and just kind of how my workflow goes, and just hopefully it will help you out as well. So we're going to start off with the terminal here. You can open up your terminal if you have Windows, and if you have the Windows for the Linux subsystem, you can open up Bash. And for Mac, you can just go to you can search for the terminal app. You can open up your terminal here, and I'm already in a directory that I want to start off in, and I'm going to increase the size here, so I'll just make this large. So first I'm going to make a directory called Coronavirus, and I'm going to change into the coronavirus directory and list, nothing in there. And then I'm going to make a few directories. I know that I'm going to want to store the software I download in a directory called software. That's where all the bioinformatics software I download is going to be located in. And then I want to have my data in a separate directory. I want to have the results that I make with the software in a separate directory. And then if I write any scripts to automate my workflow, I'm going to create another directory called scripts. Make directory, list, and then I have my data directory, results directory, scripts, and software. So we're going to start off in the software directory, and this is where I want to download Muscle and Hammer. So first, let's go ahead and go here and let's download, let's do Muscle first. So Muscle download, Muscle alignment download. So we have this first link. If I type in Muscle alignment download, I have this first link I'm going to click on here. And I'm going to do two different options here. I'm going to download I'm going to go through downloading the binary file, and then I'm going to do the source code as well. So, okay, so I have a Linux operating system, so I'm going to download, and I know it's 64-bit. If you have 32-bit, or if you have Windows, or if you have Mac, then you would do the Darwin 62 or 34, and it's the same process, but you just download a different file here. For Windows users who are using Bash, I did download this before on my past Windows computer, and I did the source code. So, which is why I'm also going to show you how we're going to do it from the source code. But first, I'm going to show you downloading the binaries, which is particularly helpful for the Mac users. So here I'm going to download 64-bit, and I'm going to copy link address here. I'm going to do wget, paste this in, download that, and then here I'm going to do tar extract file, and then muscle list, and I can get rid of the tar file here, and then now I should be able to just straight up run the file, and it works just like that. That's it. I can use it. I don't need to do anything extra, and don't even need to make it executable. It's already ready because you just got the executable files, right? Didn't have to do anything extra. Now, and it's the same process for for Mac users. Okay, you just download that binary, untar it, and once you untar, go ahead and you can execute it. Okay, I haven't showed you how to use it from anywhere yet, but I will cover that later. Next, we can download the source code. So source code here. I'm going to click on the source code. It's going to automatically start downloading into my downloads folder. I want to move it into this directory here in my software folder. So I'm going to just drag. I'm going to drag this here. Oh man. Oh, looks like I'm going to have to be moving this. Go into my downloads list. Yeah, so I want to move this folder. I'm going to move this file here, so we'll do move this, and we'll start off in my home, and then we'll do document bioinformatics class, and then in my bioinformatics class I'm going to go coronavirus, and the path might be different for you, and then I'm going to go software. Move it there. Clear, change directories, home, documents, bioinformatics class, Chronovirus software. And then I should have my this file here, this is what I want to untar now. So we'll do tar file muscle, and then so we can remove the source file now, and then now the source file that we just downloaded is actually a folder, and within that folder there's another folder called source, and within that folder there's all this all these other files, and then we can see if there's a readme here. So we can use less readme, and then so source code for readme, here's a readme. It kind of tells you some information. So we need to use the make file, and just need to run make install. Right, and I don't. Let's see. Yeah, so I don't think there's an install file here, just a readme. And then, so we have the make file here that we're going to be using to actually compile. So it looks like it's just C++. It's written in C++, and I said that in the readme text. So now all we're going to do is just run the make, and then we can actually look at make as well. Alright, it makes it just a bash script here, and it's going to compile the program into, it's just going to compile muscle. So to do that, all we do is /make and click run. And click enter, and then it's going to start compiling. It shouldn't take too long. Okay, so type in ls, and then here we have muscle now. So we can actually run muscle, and it works. And that's how, if you have a Linux, or if you have the Windows subsystem and you have bash, you would do those steps that I just did: download the source code, untar the source code folder, go into the source directory, and then just run ./mk. Okay, so now we're going to move on to Hammer. So we have, okay, so actually I'm going to remove, before I get going, I just want, this is just demonstration purposes, but I'm going to remove the muscle command. Or let me just show you real quick. Let me go to muscle and source, move muscle back one directory, this muscle, and then we can move muscle again. We can move muscle out. Oh, move muscle back one directory. And then now we have muscle here, and then we can still type /muscle, and it still runs. Okay, so that's what you can do if you don't want muscle to be inside that bin, inside this folder anymore. You can just move it into the software. But I'm going to remove the muscle 3.8.3.1 directory, and then I'm going to remove muscle. And then I'm going to move muscle here to just muscle. I did just rename it. It's just the binaries, so here it is. Okay, so we're going to continue on. We're going to download Hammer now. We have muscle ready. Now we're going to download Hammer. So Hammer download. So we go to hammer.org, you can go to download, and then we'll download the source file here. Oh, let me see if it'll let me do copy link address, wget, paste, and wait for it to download. And then same thing, but this time, because we download the source code, we actually have to compile it into the binaries. So we'll have to untar, just like we did in the muscle. File, hammer, untar, and then we can remove the .tar. Don't need that anymore. And then we go to hammer. And then here we can look at the README. Right here, README to download and build the current source code release. wget, we just did that. We untarred it, changed directories, and then we can run .configure -- prefix and then your install path. So this is important. When we do .configure, and we add the and we add this option -- prefix, we want to put the path to, I'm going to put the path to my software directory that I made. So .configure -- prefix, and then I want to do home Jesse, and then once I do Jesse, documents bioinformatics class, Chronovirus software. So this is exactly where I want Hammer to be downloaded into. So I'll click enter. All right, type in ls here. Let's run this, and it says make check and then make install. But we can also look at the README or the install as well. It tells you to run .configure, and then we run make check, and make install. If you looked at the install first and you run .configure, you wouldn't have known. If you looked at the README, you would have saw that you have that prefix option right here. Okay, then we'll do make check, and make install. Okay, so while that's going, we're going to go ahead and move on. While that's downloading, I'm going to download the genome, and I'm going to download the proteins. So first, we need to download the human coronavirus protein. Hammer is being installed right now. So coronavirus genome. Here's the entire URL. I'm going to just do this by hand. So ncbi. Center, and then we'll do, should be nucleotide, and then here's, I'm going to copy and paste this number into there, press search, and here it is. We have the severe acute respiratory syndrome, coronavirus 2 isolate, complete genome, and if you want, you can read exactly. You can read it. You can read more about it, about the journal where it's published, more about the actual where it was extracted from, where the virus was cultured, or wherever they got it from, some seafood market. Okay, but we want the genome though, so we're going to go to FASTA, send to file. We want FASTA format, create file. Okay, so we need to run make. So with Hammer, I'm actually going to complete that in a little bit, but let me go back and just get this set up here. So I'm going to take this. So this right here, the sequence.fasta in my downloads file is in my it's in my downloads folder, and I want to move it to my data directory that's in my coronavirus project folder. So we'll move this file here to data. And then if I change directories into data, here it is. And I'm gonna, oh, you know what? I made a mistake. I don't want the sequence.fast. I don't want the genome itself. I want the proteins. Yeah, protein set. That's my bad. I made a mistake there, but that's okay. And it's not too hard to fix that. So I'll just go back to nucleotide, I'll just do the same search. And I want the proteins. Let's just click here to the severe acute respiratory syndrome. If it has the complete proteins, it should have the Oh, here it is, protein. Should be more. Actually, it is a virus. Should be a little bit shorter or smaller. Okay, well, we'll just go with protein right here. Are these all from the same? I hope it is. I think it is. Alright, well, we'll just go with this. We will send to file FASTA create file. Okay, so we have this new sequence here, so I'm going to remove sequence.fasta, nothing in here, and then same thing, oh, I don't know, oh, there it is, move here, list, yeah, make sure it's protein. Alright, so we have these proteins here, and we will human coronavirus. I just want to get this. Yeah, okay, so it's from the same, just wanna, I just like gotta double check sometimes. Okay, so we have the human coronavirus proteins. And I'll go to software here, and then where I left off with muscle is I just need to do make check. Oh, not muscle, hammer. Make check. Okay. So make check is being run. Okay, so now just to check off, we got the human coronavirus protein set here, and now we need the replicase polyprotein 1AB. So we're gonna be using Uniprot KB, which is a protein database that has really rich annotations for proteins, and also scores proteins based off the annotations. So let's see if I can search for any. Okay, so here it is. So uniprotkb.org. Now we got to search. I'm just going to go straight for advanced search, and I'm going to type in protein name, replicase polyprotein 1AB. An organism, bat coronavirus. And then can we automatically do reviewed? Yes, we can. And we're going to click reviewed. Okay, and search and see what we get here. Here they are. So we click the reviewed, and just to show you real quick, if I click on the entry right here on the results, Annotation score, when they're reviewed, usually it's high annotation score, protein inferred from homology, so the protein is similar to some other protein that's probably annotated at maybe experimental level, hopefully. So this indicates the type of evidence that supports the existence of the protein. Yeah, it doesn't give accuracy. This is inferred from homology. The best thing it would be, yeah, experimental level. You want experimental evidence at protein level, and you can read more information about here. So it says that experimental evidence at protein level indicates that there's a clear experimental evidence for the existence of the protein. So so this could be Edmund sequencing, mass spectrometry, X-ray, NMR, good quality protein interaction, and then this is what you would probably want in all of them. But it's okay if we have protein inferred by homology. Don't have to be. This will suffice. High annotation score. So what we're going to do is we're going to download these. You can also add these to the basket. And then later, if you want to come back to them, it'll still be added to your basket. I think the website actually somehow stores it in your browser, the information that you have, in regards to what you have in the basket. So we'll download here. Download, and we're going to do download all. We're going to do uncompressed. I don't want to entire. Okay, so we'll do uncompressed. Click go. And then same thing here. We'll just hold off with hammer. Go straight into the data directory. So we'll do move this whole thing straight into this directory. Now we have that file here. Let me list. Here it is. So we have this uniprot, your list, this long named file. We can do head uniprot, and you can see that it's the bat replicase proteins. And let's go ahead and let's rename this to bat replicase proteins dot FASTA. So now we have our bat replicase, and I want to change this as well. I want to put a underscore in between. Okay, so we have the bat replicase proteins, and we have the human coronavirus proteins. So bat replicase proteins, the first one we just looked at it, but I always like to ensure that I have an actual FASTA file. This is really small. Okay, so we'll go ahead, clear, go back, and back to our software. So we just need to finish off with hammer here. And we did make check. Clicking up arrow, we can go back to our previous commands. I think our last make command was make check, if I recall. Yeah, make check. So we'll just do make install now. And then make install is good to go. Now we can go back to our software, and now we have new folders. We have share, and we have bin. So we go into bin. We have our hammer command, so we do /hammer or HMM build. It works, it works. We can do a HMM search. Yeah, nice. Okay, now what we would like to do is kind of work in our coronavirus directory, kind of do all use our software in this directory and kind of put the results and put all the results into the results folder. But if I try to use muscle from here, I can't. It says no such file or directory, right? Or if I do HMM build, the files I'm getting no such file or whatever. Or if I type in HMM build, command not found, right? I want to be able to use this command from any directory essentially. And the first way to do this is that we can just go straight to the software folder, excuse me, and then we can look at the environment variable path. So we can echo out the environment variable path here just to take a look at it. And what you'll see is that you'll have these absolute paths starting from home all the way to whatever folder it is, or right here, bin. And what we need to do is we need to add the software folder to our environment variable path. So we need to add it to this path here. So right now, as you look, we have /home/Jesse /.local/.bin, and then within this folder here, it maybe has some commands here, and then this colon right here separates and means that there's a new / here, which means this is a new directory, home Jesse bin, and then within this bin there might be other commands. And then it goes /user /local /s bin, which means there's a whole other commands that we can use. And in those commands, when I type in ls here, it's going to look into each absolute path here that we have in our path, and it's going to look for ls within that, in each absolute path here. So, and same thing if I type in cd and I press enter, it's going to look for cd in all these absolute paths. It's going to go to home Jesse /local bin. It's going to get the bin right here. Look in that folder. Is there a command or a program called cd in that folder? If there is, call it, run it, and it works. If not, look in the next absolute path. Look into this bin folder. Look into this one. If it doesn't find it, then it runs, and then it tells you command not found, just like how we did with hammer build, HMM build, and then command not found. What happens is that your shell will look into and try to search for the file called HMM build, and it will look for it in that folder. And if it can't find it, or actually it'll look for it in all these different folders they have here, every single one of them. And if it finds it, and then it will run it. If it doesn't, command not found. What we want to do is add our full path, the absolute path of my software, or wherever you download your software for this little project here. I want to add this whole thing to this variable, to my environment variable here. I want to add it on. I want to put a colon at the end of this one, and I want to add it over there. Okay, and actually, looks like it's almost already, it's already there in this one. But here, if I type in muscle, I go here. If I type in muscle, MUSCLE, right, it's not working. Okay, now what I need to do is I need to say, I just need to recreate it. I'll use export to create a new environment variable, and I'll just call it path. So I'll be recreating this variable here, and I will put double quotes, and I'll put path here. And what this does is we'll just expand. So now it's going to be the same thing. I'm just putting this whole thing into here, which will equal path, and nothing new there. It's going to be the same what it's already set to. But now I want to add my current path working directory. So I press enter here, export, and then now if I do echo path, and then here we have this new one, home Jesse documents bioinformatics class Chronovirus /software. Now, if I type in muscle, it works because I added the absolute path of software. So I add the full path to the folder called software, which contains the command or the software muscle. And when I type in muscle now, it will be able to find it in the software folder because I added that to my absolute path. Okay, but one thing to note here is that when I exit out and I open up this again and I type in muscle, command is not found, and that's because I manually typed in export path equals, and then I said path, and then I did whatever my current working directory, my path working directory was, right? And I would have to I would have to do this every single time if I wanted to be able to use muscle that's located in my software folder, right? And to make this work, we can have it so that this command that we ran here is ran every time that we open up the terminal. So it kind of just does it for us instead of us manually doing it. And all we need to do is modify an RC file. So if you have bash, it would be .bash rc. So you can use your text editor like Nano or Vim, okay? And you just do .bash rc. If you were using Windows and you're using the bash terminal, you would open up your .bash rc, and at the very end here, you would just say export at the very end of the file. You would do export path equals, and then we would do path, and then we would do the absolute path to the folder that we just created. So home, Jesse, and then within home and Jesse, there's documents, and this path might be different for you. You can just use the pwd command to see what the absolute path is if you forget of that folder. And then I went to bioinformatics class, and then from there I went to Chronovirus and software. Okay, so software, and here it is. So here's the full path that I would put. Okay, so same thing, export. So we're going to create a variable called path. You can actually just ignore this right here, but what we're going to do is create a variable called path here. Okay, and then all we're going to do is substitute. We're going to take what the current variable called path equals, and we're going to, or what it's assigned to, and we're going to expand it all the way, and then we're going to add a new path, a new absolute path to our variable path, and we're going to add the absolute path straight to software. And remember, this folder here contains muscle in it. If you look into it, it contains muscle. And then if you have I'm going to go ahead and, if you have Mac, one thing I forgot, and I apologize, you need to first go to your home directory and then type in, then you can access your .bash rc file, and then for Mac users it's .bash_profile. Okay, and for me, I'm using a different shell, but it's pretty much the same thing. So what I'm going to do is I'm going to take what I wrote in my bash rc file, and I'm going to just copy it over into my Z shell RC file. So here, all I did was just, I have my bash RC, my .zsol.rc file, and it's same thing I put in my bash rc file. All I'm doing is just putting the full path, adding the absolute path to the software folder, to my environment variable path. And then now, every time if I exit out now, and I open up terminal, if I echo out what the environment variable path equals now at the very end, it will have home, Jesse, documents, polyprotein class, which is what I added, right? And I just type in muscle. And it works. Now, do I have hammer? No, I do not. So, let's change directories into the software folder. And then, so here, hammer, the commands are located in bin. Right, it's located in the bin folder, not in the software folder. So if you type in pwd, then this will give me the absolute path all the way to bin, which inside the bin folder contains all these commands that I want to use from hammer. So to add this to my path, I can just repeat what I did earlier, is go back to home, make sure you type in cd, enter, and then with your text editor, such as nano or vim, open up your bash rc file or your bash underscore profile file, and you're going to edit the last line here. And essentially, you could just add on bin. Okay, copy right here, and then paste it at the bottom, underneath it, or above it, and then as /bin. So now when I type in muscle, it will look for muscle, and once it gets to this folder right here, it will go to home, Jesse, documents, bioinformatics class, coronavirus, and it will go to software, and then it will actually find muscle within that folder. And then for hammer, it will go into software, it won't find it in that folder, then it'll look inside bin, and it will find the hammer commands like hammer build, hammer search, quit, and then just to show as an example that this works, HMM build, and it works. At HMM search, and it works from anywhere now. I just type in the command, and it will now my shell will be able to search for hammer, find it, and then run it, because I added the absolute path of where hammer is located, which is inside the bin directory that's inside my software. As I showed you, it has that whole absolute path. Now the shell knows where it's at, and because I added the export command, where I recreate the environment variable path again with the new absolute paths added to it, added that to my RC file. Every time I open up my terminal, automatically creates the new path variable for me. I don't have to manually do it myself. Okay, hopefully I made sense there. Next, we're going to go to documents, bioinformatics class, coronavirus, software, and then we'll do, now we just got to run muscle here. And now we can actually do the calculation. So now we can use muscle, right, and we can use hammer, the hammer commands. So we're good to go. Now it's just calculations at this point. In my next video, I'm actually going to show you an easier way to actually download all this. But just want to get you an idea of downloading some software by hand, because sometimes you may have to do that. So we don't need this anymore, don't need this, don't need this. Next is meeting our objective, which is searching for the replicase proteins. So first, what we want to do is we want to use muscle to create an alignment. So you can just type in muscle and press enter, and it will kind of give you some output if you don't give any arguments to it. and it tells you how to use it. Basic usage, muscle in. So we use the -in option is the file that we're going to give to it, and then we have the -out option, which is going to be the file that we're going to, the name of the output, so the multiple sequence alignment. And then here it says you can just read more about the options, and then the other options that they have here, and it says that it gives a input, it's an input file. When we use -in, it has to be FASTA format, and then out file, output alignment in FASTA format. So let's go ahead, muscle, and remember we put the data, and we're going to take the BAT, we're going to align the BAT proteins first, and then we're going to do out, and we're going to output the result in the results folder. We'll do results, BAT, replicase.aln, and then we'll click enter here, and then it's going, muscle is going to align the proteins and create a multiple sequence alignment for us. Okay, so we have the results back now. We go into results, and we can actually look at the alignment file here. And it's just like the command said when we press muscle and enter, it kind of told us what the output's going to be. The output is going to be in FASTA format. So we have the greater than symbol here, and then we actually have the header of the sequence that was aligned here, and then underneath it is the actual sequence, and then you see these gaps, which means that the sequence has some insertions or deletions, and we can actually scroll down and look for another header here. So here's a different header, different sequence, and then so the sequence is aligned, and we can now use it. We can now use this alignment in Hammer to build a profile hidden Markov model. So we're going to use HMM build. So again, if I just say I don't know how to use I forgot how to use this command, so I just type in HMM build, press enter, and then because software is pretty user friendly, at the command line it tells us how to actually use it. Usage, we just type in HMM build. There's some options that we don't have to put, but the main part is the name of the file that we want to create. So the Hammer file output, we can name it whatever we want, and then we can also give the multiple sequence alignment file here, and then also have some other basic options. And then if you want to see more, just type in HMM build -h. So we're going to go ahead, HMM build, Hammer file out, so we'll just say BAT replicase.html. And then we will say, oh, I actually want to put this in my results. Results BAT replicase.html. And then the from the past results with the alignment, BAT replicase.aln, press enter, and then now HMM will build our profile hidden Markov model. So we can just take a look at the first few lines here. Well, might as well just look at the whole thing. Okay, so here it creates the the matrix here that it's going to use to search and score the sequences in the human genome, the human coronavirus protein set that we downloaded. So now we have our HMM model that we're going to use now. We're going to use this model, take it, and we're going to use it to try to find other replicase proteins, replicase polyprotein 1AB in the human coronavirus protein set that we got from NCBI. Okay, so we might get results, we might not. We're about to find out. So we'll do HMM build. Oh, not build, search. Same thing, we just press enter, it tells us how to use it, HMM search. Then we use the hidden Markov model, or the model that we built right here. And then we give the sequence database. The sequence database is the protein set that we downloaded, the human coronavirus protein set. So what we do is we'll just say H build results bat replicase.hm, and then we'll do data human coronavirus. Press enter. Let's see what happened here. So there was while reading a line that FASTA file, .fasta, there was an error. The sequence has a length of 4,405. What is this? YP00. Is it because of those spaces? Oh, I just moved this space there. And then we run this, see if it gives us a different error. Oh yeah, it does. I'll do it one more time. Then results that pen data human coronavirus. Okay, so I removed the first line from here just to check. Here I gave some error, remove this, and then I can remove this here. And then I'm guessing it'll be ORF10 or YP009255. So I should get this one here, YP00972 or 97.2 at the very end. So if I run there's not that many sequences here. Oh my. Oh, this is totally, I did something down here. Okay, I don't know what I was thinking there, but it's not too hard to fix. I just wasn't using the right command. Make sure I didn't mess up anything. Okay. So what I'll do here is I'll rerun the command here. I'll do the build first. I'll do the build that I did earlier. Result HMM build results is going to be the bat replicase HMM, and then we'll do results / bat replicase alignment. Press enter. Creates the alignment, and then I don't know what I was thinking earlier, but we'll do HMM search, and then we'll use the result from the past that we just created, HMM, and then we give the data genome human. There it is. Now it runs, and if I run that again, you'll notice that it just gives it straight to the actual standard output, just right to the screen. It doesn't actually write it anywhere. What we need to do is redirect it with this greater than symbol. So we'll use this greater than symbol to redirect all of this stuff printed out to the screen. We're going to redirect it, and we'll call this, we'll put it into the results file, and then hammer search results.txt. Take some time, and then we just need to, less hammer, or what is it, results, hammer search results. And it looks like we look at the top here, we have query, we have the bat replicase, and then scores for complete sequences. So we actually have two sequences here that got a hit here. We have this ORF1AB polyprotein, and then it just looks like just one protein actually. ORF1AB, and then we have ORF1A. So it is two different proteins, two different accession numbers here, sequence. And so we got two results back. We're not going to really investigate it. You can take a look at it if you want, but I just wanted to show downloading the software, using the software, and then here and there I mess up, but eventually just gotta think and figure it out. It could be a dumb mistake like I was doing, or it could be a more subtle mistake that you don't notice. Okay, so now right before I end this, I'm going to create a script to kind of automate this, so I don't have to, if I get different data, I don't want to have to keep on using all those commands, typing them in. I just want to create a bash script that's going to execute the commands I just used line by line, so I don't have to repeat myself if I get different data. If I use different data, so we'll do scripts. You can use nano or vim. I'm use vim, and we'll do scripts, and then we'll do unhammer.sh, and then we'll do the shebang here. Then you can use bash, and then we'll create two variables. The first variable will be the query, and we'll expect the user to give us the query, which will be the first argument. So the usage would be like forward slash use, and then dot forward slash runhammer.sh, and then the person would put the query here, the user, and then the database. Okay, I'm not going to add any checking or anything right here. You can modify this script here yourself and get it more user-friendly. But I just want to show you real quick how this might work. So we'll have export query and then export database. So this case, we expect this to be a fastifier. And then database FASTA file as well. And we'll do, so the query for us was the sequences we gathered from UniProcKB, which is the bat proteins. And then we first use muscle. And then we will give the query and then dash out. The output is going to be the query, same name, but we'll just add .aln. And I'm just going to put brackets here. And then after we create the multiple sequence alignment, we use build, which we will create a profile hidden Markov model with the query alignment, the multiple sequence alignment that we made here. And we can, the output, or actually I'm making a mistake already, .HMM. We create the profile hidden Markov model, and then the output will be create it. This is the output, and then our input is the alignment. So build, taken the hidden Markov model that we made here, and then we will create this hidden Markov model with this alignment, and then we will do search. And the search will be, we will search with the query.hm that we made here, and the output in the database, we're going to be using the database, the searching. So with this model here, search in the database, and we will redirect the output as just results. We'll call it runhammer results.txt. Okay, and then we can add some comments as well, create alignment, just to make things more clear for us. Create alignment. Build with alignment. And then search with and database. And so that should be pretty clear. We create the alignment first, build the hidden Markov model with the alignment, and then with the alignment we search in the database. So with hidden Markov model, search the database. And then we will make the command executable. So we'll do chmod. We'll make the script executable. And we will test Okay, so now scripts, run hammer, and then now the first argument needs to be the query. So from the data, we'll use the bat replicase proteins, and then from the data, we will take the human coronavirus. Press enter, and then just wait for the results to come back. So it's doing the multiple signals on it first with muscle. And then it's creating the profile hidden Markov model. And then it should have searched. So we have the results here. So we get less hammer results, and then here it is. We have the two proteins here, ORF, the open reading frame AB, and ORF1A. And that's pretty much it. That's the end of this video.